In this experiment, we have three objectives. So the first one, to calculate and describe the following destructive statistic: mean, variance, and standard deviation. The second is to understand the concept of the correlation. And then the lastly objective is to observe the part of the leaf or plant. In this particular one, we have the four material, which is the leaf, ruler, paper, graph paper, and then the lastly is the weight balance. This part, I will explain about the step by step about the procedure in the practical one. The first step is the 10 leaf were taken, measure and record their length and with the mean, variance and standard deviation of the length and the width of the 10 leaf were calculated. The data was tabulated in the graph paper. So the second step is the another 10 leaf were taken and repeat as the above. It was done for the another 3 set of the leaf. The result was tabulated as the data matrix. So the lastly, all the data of 13 leaf were used to plot a graph of the leaf length versus the leaf width.
Let's explain about the results of the lab 1, which is where you guys need to know we start with the set 1. Set 1 from the quarter 1, where we have to check the leaf length, where the mean here is 4.360. Mean we get from the table above there, where we divided by the 10 number of leaf and followed by the variance is 0.243, followed by the last one is the duration, where the answer is 0.493. One here is the leaf weight, where you guys need to see the table here of the 10 leaf, where the mean here is 17.350, followed by the variance is the 3.868, and followed by the last one, the duration 1.967. For the third part here, which is leaf weight, same as before, we need to find the mean, the variance, and also the last one is the deviations. So the mean here is 0.864, we get from the table above. Same, you guys know how to find the mean, right? So for the variance is 0.0333, and the standard deviation here is 0.1825.
So we will move on to the set 3. So based on the set 3, we will look at the three things same as before, which is the leaf length, leaf width, and also the leaf weight. But we will only focus on the leaf length and also the leaf weight. So for the leaf length, based on the 10 leaf that we have collected, we can get the mean, variance, and standard deviation, same as the set 1 and also set 2. But for set 3, the mean is 11.110, and then the variance is 3.865, and the standard deviation is 1.966. And then we will look at the leaf width for set 3. So for set 3, same as we got from the leaf length, we will look at the mean, variance, and also standard deviation. So for the mean of the leaf width, we got 0.298, and then for the variance, we got 0.375, and the standard deviation is 0.612. And lastly, for set 3, we look at the leaf width. So same as leaf length and also leaf width, we just use the same calculation for mean, variance, and also standard deviation. So for the mean of the leaf width for set 3, we got 0.031, and for the variance, we got 0.099, and for the standard deviation, we got 0.315.
Discussion. Normal distribution is defined as a continuous, symmetric, bell-shaped distribution of a variables. Since the normal distribution is simply a theoretical distribution, there won't be any variables that exactly fit this distribution. The variance, standard deviation, and mean are some of commonly used parameters for a normal distribution. The average data of central tendency is called the mean. Next, the variance, denoted by the symbol sigma squared, represent the statistical dispersion about the mean among the numbers in a data collection, which means that it measures the distance between each dispersed number in data set from the mean. Meanwhile, the standard deviation, denoted with the symbol sigma, is the square root of the variance which helps determine the consistency of the data value over a period of time. In this experiment, 40 pieces of leaves with random length and weight were divided into a set of tens. Each leaf length, width, and weight were measured in order to get the data about for each leaf. A graph of leaf length versus leaf width was plotted. For each scale, values such as the mean, variance, and standard deviation were calculated. Correlation is a statistical method used to determine whether a relationship between variables exists. Based on the plotted graph, the relationship between the length and the width of leaf was determined. The graph shows that there is a relationship between length and width of leaf, which is a linear relationship. The scatter plot can be seen to have a positive linear correlation, which means that leaf length proportional to the leaf width. In this case, when the leaf length increase, the leaf width also increase. So we can conclude that both variables are dependent.
Conclusion, we learn how to determine the calculate and standard deviation, variance, and mean from this experiment. We can say that the one increase with the length, which makes the variable are dependent to each other. The first question is, discuss the differences or similarity of the mean of length between sets of leaves. Do the same for the mean of width. We can say that the mean length and mean width different amongst the leaf sets. Still, those settings doesn't differ all that much. The edge of each individual leaves may be the source of discrepancy. The leaves' identical looks indicate that they belong to the same species. As a leaf ages, its size will be increased. Consequently, a leaf size decreases with age. 5 sets of 10 leaves are randomly selected. It was group similarity in size, so there was a little difference between the mean and individual values. Next question, what is meant by variance? The leaves have different measurements of width and length. What is the meaning of this statement? Discuss with respect to variance. What is meant by standard deviation? So the answer will be the amount that the data deviate from the mean value is measured by variance. The statement demonstrates how a set of leaves can vary in length and width. No individual in a population is alike, regardless of size or variance. The variance, however, indicate that there aren't many significant differences between the size. So the tiny value of the estimated variance indicate that the leaves' length and width did not deviate much from their mean values. This is due to the fact that the data distribution spread from the mean value decreases with decreasing variance. Okay, as for the last question is, what is necessary to have different set of measurements for the measurement of leaf width and length? Okay, it is necessary to have different sets of measurements of leaf and width and length because to make sure to get a consistent outcome. If only one measurement was used, it is difficult to evaluate whether the result is accurate. So when we have different sets of measurements, we can compare our result with other results to get a consistent result. It's also follow a natural law because there is no similar individual in the group. What are the objectives of this practical tool? There are 4 objectives here, which are, the first one is to introduce the simple concept of quadrat, and the second one here is to enhance the power of observations, and the third one is to record the difference in turn distribution within the quadrat, and the last one is to infer from natural observation the possible factors that affect the distributions and the abundance of organisms. So, for the introductions, all living things found in environments are considered to be dysbiotic components. A population is a collection of organisms that belong to the same species that are present in the same habitat. A community is made up of populations of several species coexisting and interacting in the same space. Making a list of every species present in the region and also contrasting the size of populations is one method for researching the relationships between organisms in a community. We shall be introduced to a basic quadrat machine in these practicals. Along with classifying the plant species present, we also need to monitor the ability aspects of a research site. So here there are 5 materials needed, the first one is measurement tape, followed by the stick and notebook here, and the raffia string, and also the last one is scissor. In this practical, forest camps regarding your item jangka Pahang have been the areas under study. 4 quadrats of 5 meter times 5 meter were made in the forest. 4 quadrat samples of each group were performed throughout the habitat at several random locations. All 3 stands found inside the quadrat were observed and results were taken. Data was used to estimate the population size and population density within the entire habitat.
Hello, I'm Kasih, and now I'm going to explain and show to you guys about our result and observation. Before I start, this is the species that we named and the symbol that are given to each species for each site. So this is the first site that we went to observe. As you can see here, there are 8 species in total exist in this site, with the total number of individual of 18. The next site that we went to observe is site 2, with 4 types of species and 42 total number of individuals exist in this site. And for the third site that we went to observe, there are 5 species exist in this area, with the total number of individuals of 8. And for site 4, there are 4 species that found in this area, and with the total number of individuals of 16.
So now we look at the post question in order for us to know or understand better about this practical. So the first question is define the term quadrat in practice. So quadrat is an ecological sampling unit consisting of small square frame that is laid down which all species of interest or specific area of the community to be sampled, and the measurements are taken. Within the quadrat frame, the existence of plants is recorded using an appropriate measure of abundance. The second question is, what are the 2 basic requirements of all the quadrat techniques? So the 2 basic requirements of all the quadrat techniques are the area of quadrat counted is known, so that the density is determined directly. Besides that, the organisms must be immobile during the counting period to ensure that they are counted and not missing. Okay, third question, what would be the advantages and disadvantages of using a larger or smaller quadrant to survey the sedentary organism? So the advantages of using a larger or smaller quadrant to survey the sedentary organism are, it is inexpensive because it only requires few materials. Besides that, it is a feasible method and easy to use compared to other sampling methods since quadrats are uniform in size, in shape, and distributed randomly throughout the sample area, which makes the study design straightforward. Apart from that, larger quadrant can give more accurate estimation of overall densities, but it can be complicated to carry out due to the time consuming to place and sampling them. Despite relative ease of designing the quadrant, they might be study areas thicker too. Quadrats they are too small or too large or spaced inappropriately, often result in errors. For example, random space quadrats which are too small might miss too many individuals to be counted, which will result in underestimation of actual population size. And other than that, inconsistency in counting and omitting spaces that lie partially in boundaries also can occur. And the fourth question is, problems with spatial scale often make it difficult to directly estimate average densities habitats, such as large forested areas or in aquatic habitats, and cannot directly see the animals or plants of interest. So we have to explain the issues concerning ecologists in conducting the sampling method. So the sampling method is relatively straightforward in that simply involves counting the number of organisms of interest in a defined area. However, there is some problem with sampling that can be thought of as several issues. Firstly, the sample method that can be used to collect the needed data. Secondly, ways on how to select sampling locations to limit the potential bias and obtain base estimates of target parameters. And lastly, ways on how to minimize the number of samples that they take while still providing an accurate estimate of parameters. So in consequence, ecologists have developed several basic sampling approaches to deal with sedentary organisms, which is the most common method, which is the blood sampling.
I want to explain about the discussion in the quadrant 1, 2, 3, and 4. So, to provide image, is show a quadrant with the various symbols representing different plant species. The table summarizes the species name symbol, total number of individuals, S, and their density in percentage. So, the distribution of the species within the quadrant is uneven. For example, species in the set 2, symbol X, is the most abundant species, which is 39 individuals, make up the 86%. Also, in the set 2, like the symbol love, flower, and the diamond are much less abundant with only one individual respectively and representing about the 2.38% of the total. So, the density of each species varies significantly. The higher density of the species symbol love might indicate that it's well adapted to the condition of the quadrant area, outcompeting other species for the resource. On the other hand, species with a lower density might be less competitive and might be specialized to the different microhabitat within the quadrant. So this practical exercise using the quadrant method help to illustrate the concept of the species distribution and density, enhance the observation skill, and allowing for the inference about the ecology interaction and the environmental influence. Understanding this pattern can provide the insight into the functioning and health of the ecosystem.
So what is predation? Predation is an interaction in which one organism, the predator, is all part of the body of other organism, which is prey. So the transfer of energy and nutrients is actually the transfer of energy and nutrients in community. Okay, there's like 3 examples of predation, which is herbivore, carnivore, and parasitism. Okay, the predators' numbers are dependent on prey, and predators may regulate number of prey. Okay, the mechanism of plant herbivore system. Defoliation and consumption of fruit and seeds. What is defoliation? So defoliation is the destruction of plant tissue, which is leaf, bark, stem, sap, and roots. Its feeding on seeds can be determined. Plant responses after defoliation. The defoliation and subsequent growth alter the tree site physiologically. It can cause the change in hormonal growth regulator that control the bud dormancy. It also can cause the increased attack by insects and diseases, and it reduces the resource available to reproduction, which is loss of nutrient and mature cells, and also in some instance in coniferous tree, the defoliation can cause death because these trees do not have physiological traits that allow them to recover. As for prey defenses, for chemical defenses, there's like 3 examples. First example is venom and pheromone. First one is snakes for venom. It's to avoid predators or capture prey. The second is fish. Sea lamprey release the pheromones that act as alarm and induce fringe and flight. As for the second example is secretion. Arthropods, amphibians, and reptiles. It takes up secretion to repel predators like birds, mammals, and other insects. Okay. Component amounts is strongly odorous secretion and easily detectable. Secretion for slimy substance and mucus may come out from the body walls for the millipedes and may be released in air for the beetles and spray as in grasshoppers and stink bugs. Next, for prey defenses is flashing coloration. Butterfly, grasshopper birds display visible colored patches when it distribute and put the predator running away. And the color may distract and disorientate the predators. In deer, the colors may sign to provide a group of organization when comforted by predators. There's one example that represents flashing coloration for prey defense, which is harmless skull king snakes. It's actually look identical with poisonous coral snakes. response. The first one is cryptic coloration. What is cryptic coloration? It is also been known as camouflage. It is the use by various prey species to blend into their surroundings and hinder predators' ability to identify or detect them. To successfully hide them, these strategies includes two things, which is pattern and shape, and also the last one is texture. How about the pattern and shape? It is the background matching. The colors and patterns of these pieces often closely mimic those of their natural surroundings, including any object there. The second one is posture. About posture, we want to explain more on the freeze response of the prey. Many prey species use their patterns and coloration to blend in when they detect danger, causing them to freeze and not moving at all. Second type of prey defense here is alarm call. What is alarm call? It is the use by prey animal uses alarm calls or other vocalization or signals to warn other animals about the presence of potential predators. There are many examples of alarm calls that use or prey that uses alarm calls as defense. But here, three of the examples here. The first one is bird, second one is honeybee, and the third one is squirrel. As for the bird here, we use chickadees as the example. It uses high pitches calls to one of the predators. Their calls vary in length and complexity depending on the type and size of the predator. Second one is honeybees as pheromones. When a honeybee stings, it releases alarm pheromones that alerts other bees to the threat. It is crucial for colonies' defense against larger predator. And the third one is squirrel as eastern grey squirrel. It's known for their vocals and visual alarm signals, which help them avoid predators.
What is the cannibalism? In zoology, cannibalism is the eating of any animal by other member of the subspecies. Cannibalism is a common ecology interaction in the animal kingdom and has been recorded in more 1,500 species. Can either be active, who hunt and kill their conspecific before eating them, or passive, who feed on the already dead member of their own species. In these slides, I will explain about type of the cannibalism. In cannibalism, both predator and prey belongs to the same species. Approximately 114 species of the animal are known to have the cannibalism tendency. Animal cannibalism can be divided into the four basic types. The first one is the sexual cannibalism. The second one is the cannibalism philia. The third one is the cannibalism introducens. And then the lastly is the survival cannibalism. The first one I will explain about the sexual cannibalism. For example, spiders are the deadliest cannibal. Female kill and consume the conspecific male before, during, or after mating. This may increase the female fertility and ensure the potential of the survival of the species. Example, black widow spider. So, the second type is the surviving cannibalism. Polar bears usually feed on the seal with their hands from the platform of the sea ice, but the melting of the glacier a result of the rising global temperature makes it difficult for them to hunt. So, this has led to the malnutrition and the starvation as the unable to build sufficient fast reserve for the winter. So, they hunt each other for the food in the desperation to survive. Filial cannibalism. Certain families of the lawless fishes exhibit filial cannibalism, where the cannibalistic parents either consume their well-brought or eat only some of the egg in the nest. This consumption of the offspring might be an adaptive strategy of the parents to maximize the lifetime reproductive success for nutrition gains and to reduce of the consequence of the bird overcrowding. The lastly of the type is the intrauterine cannibalism. Same tiger shark eggs develop and hatch inside their mother oviducts at the different time. When the hatcher run out of the yolk from their own egg, they eat the other eggs and hatch until the baby shark from the each oviduct remains.
Symbiotic. Two organisms live in close physical association. Both derive benefits. At least one member of the pair cannot lead an independent life. Non-symbiotic. The species do not live together. Both members benefit each other. The relationship is either facultative or opportunistic. Example, best seen between the pollinators and angiospermic plants and in dispersal of roots and seed by animals, including birds and mammals. In such type of association, the two species remain physically unconnected. Mycorrhiza is association between fungus and plants via the plant root. Both organism, plant and fungus get benefit from each other. For example, the plant supplies energy from photosynthesis to fungi, and the fungal hyphae take up mineral nutrients from soil and transport them into plant's root.
And the second one is defensive mutualism. In defensive mutualism, one partner receives food and shelter, and in return, it helps the partner by defending against the herbivores or predators or maybe parasites. The example of this is oxpeckers and large herbivores. And oxpeckers are birds that perch on large herbivores like buffalo or rhinoceros and eat ticks and other parasites from their skin. And then the herbivores benefit from the parasite removal, while the oxpeckers gain food source. The birds do not stay with the herbivores permanently, however.
And for the last one, I'm going to explain to you guys what functional response. Functional response is a relationship between prey and predators, and there are 3 types of functional response, that is type 1, type 2, and type 3. The first type of functional response is type 1. The type 1 graph shows that the amount of food of a predator consumed increased linearly with the food density. We assume that the food processing time is negligible, in which it means that the time for the consumer to prepare and eat its food does not interfere with the search of food. However, once it reaches to its maximum and reach the constant value, this indicates that the predators are at satiated, and the prey population will stay in constant value. And the best demonstration for this type 1 graph is the long-eared owl and the microtus vole population. And now for the type 2 functional response. A type 2 functional response curve is shown here on this graph. As the density of prey population increase, and you will start to see a curve plateau. This plateau is thought to be due to the time it takes for the prey to eat and process its prey, interfering with the ability to search for more prey. So as the density of prey increase, it's already eating as many prey as possible and cannot search for any more. The best demonstration for this type 2 functional response is amplified crustacean, that is corophium, being eaten by a shorebird, that is Stringatodonus. And for the last type of functional response is type 3 functional response. The type 3 functional response is similar to type 2 functional response, in which one was high level of prey consumed, there is a plateau. However, type 3 functional response can be distinguished by its accelerating relationship between the density of population and the number of prey consumed. And this explanation graph increased because the predator has developed and learned a new skill to search and attacking for a particular food item. And the best demonstration for this type 3 functional response is the bay-breasted warbler and feeding on a spruce bloodworm. And this graph shows all the type of predator functional response for type 1, type 2, and type 3 functional response.